better.
Since
Now we
I'm
just
jumping in for Lucien at the kind of at the 11th hour, which I'm happy to do, but big shoes to fill. So anyway, yes, so I'm going to share some highlights
Oops.
from the International Research Congress, which was held just, boy, a week and a half ago in Amsterdam. And for those of you who don't know, the society has held this research congress, kind of a meeting of scientists in the field for 32 years. Started as a very small meeting. It was only in the US up until 2019 when we decided to take the leap across the ocean. Um, Partly because, we always knew that FSHD is universal. Everybody, people around the world have it. And also many of the top researchers in the field are actually in Europe, and the society has funded them. And and we know that, it seemed only fair, like it didn't seem fair to make all the Europeans travel to the US every time. And then we kind of divide up the travel. And it's been really exciting because actually there are a lot of young researchers in Europe that don't come to the US for our meeting when it's in the US, but they throng to the European meeting, and we had record attendance. We had 240 people, I think, attend in Amsterdam, and our previous record was 180. So that shows you this enormous boom. I think that bodes well for the FSHD field. So, and Amsterdam was a great place to meet. So I just wanted to quickly give you like a big picture overview of how research is, kind of how we think about research. It starts with what we call preclinical. That's before it goes into humans and patients. A lot of work in the lab, test tubes, all that. Then you start to study in human beings. It's called clinical research, like, what can you see on an imaging device, et cetera. It's just to understand how FSHD manifests in actual people and human populations. And that's really critical, because if you don't know that, you cannot develop treatments, because you don't know what you're looking at when you give treatment to a group of patients. What are you going to measure, and is this just part of normal variation, or is it actually something related to the treatment that you're giving? So clinical research is critical. Then we have clinical trials where you have an intervention. It could be anything from physical therapy and psychotherapy to a drug, to all whatever you think will improve the condition or alter its development and trajectory. And then once you get through clinical trials, you have a launch of the treatment onto the market. And then you have a whole host of other questions that you need to think about, such as access to treatment. I think probably many of you had experiences with running into doctors who really don't understand FSHD. Maybe it took you lots of years to get diagnosed. How can we shorten that time frame? How can we improve the quality of care? How can we raise awareness among clinicians? So we kind of think of these big baskets. And what I put here is a number of talks we decided to highlight that and where they fit on this spectrum. So so I'm going to proceed with I'm just going to go through the talks that we thought were really interesting and standouts from the IRC. So the first here is by one of the giants in the genetics of FSHD, and he presented his data from a study of FSHD in sub-Saharan Africans. So with a lot of the research having been done in the US and Europe, we all knew that the population, the demographics of FSHD was not really representative of ethnic diversity, racial diversity around the world. And so this was one of the first studies to look at sub-Saharan African populations. It's challenging because I think the rate of diagnosis in these areas is even lower than it is here, because people have less access to the type of healthcare that would allow for a diagnosis to be made. So, so that's a challenge, but they did find probably about a dozen, 15 to 20 individuals of African background in Africa and also in Europe and the US. And what was interesting is so FSHD is quite complex, and you have some of you may have heard about this D4Z4 region that gets shrunk when you have FSHD, and it allows this bad gene called DUX4 to get expressed, and that damages your muscles. But that only happens if you have another gene, or it's called a haplotype, a variant. There's two types, polyQ-A and type A and type B. And if you have type B, you don't get FSHD because it doesn't allow the DUX4 gene to be stably expressed. If you have the type A, though, that plus the reduction of the D4Z4 regions makes you vulnerable to having FSHD. So I don't know if you've followed that, but in the US, the proportion of people who have type A and type B is about 50-50. In Africa, interestingly, there is a lot more, like 85 to 90% have type A, which makes you think, well, maybe they should have a very high rate of FSHD, but it seems like so far that they do not. So that leads to this hypothesis: is there something in people of European ancestry that makes them more vulnerable, some genetic quirk or something that makes them more vulnerable to getting FSHD, or something, conversely, something protecting Africans from developing FSHD? So that was a very interesting talk. Obviously, more work to be done there. Next talk I wanted to feature is by Beatrice Biferali from Italy. And there she looked at the role of inflammation in FSHD. So, the muscles, the tissue does show signs of inflammatory activity. And what they found is there's a particular pathway involving something called interferon gamma. And this is well studied. It's involved in many other things, including autoimmune disease. And so the idea here is if you can tamp down interferon gamma activity, you might be able to improve FSHD symptoms. And in fact, they showed this to be the case in a mouse model. And what's exciting here is that there are a number of different drugs on the market for other conditions like rheumatoid arthritis and so on, that are already being used to suppress interferon gamma. So the idea here is maybe these are drugs that should be tested in the future in patients. Our next presentation that I picked is by a company called Scholar Rock that is getting into the FSHD space. They currently have a drug that they're testing for spinal muscular atrophy. It's called SRK-015, and it's been shown to improve muscle mass and strength and endurance in a mouse model called FlexDux4. What this molecule does is it actually counteracts or blocks myostatin, which is a naturally occurring protein that whose job is actually to prevent muscles from growing. So if you think of muscles as a very dynamic tissue, if you like work out a lot, your muscles should grow. And if you're not, and you're sitting around and being a couch potato, your muscles will shrink. So muscles are very dynamic, and myostatin is a naturally occurring protein that prevents muscles from getting too big. In fact, people and animals that have mutations in myostatin signaling will get very huge. They look like the Incredible Hulk, or very big muscles. So normally, you don't want.
I don't think you can.
Can you hear me okay? All right. So normally you don't want, so myostatin kind of keeps muscles at the right size, but in a muscle disease, you might actually want to take away the myostatin and give the muscles an extra boost and be able to grow stronger. So this is actually a mechanism that was used by Acceleron a number of years ago for a drug called ACE-83 that some of you might have participated in their clinical trial. It's also the same mechanism used in Roche's current maneuver trial. And each of these tries a slightly different tack to tackle myostatin. And what's different about SRK-015 is that they're going after what's called a precursor to myostatin. So the proteins in our body are all like, if you think of a little factory with conveyor belt with proteins being assembled, and at the end, like a full-blown mature protein comes out. Myostatin goes through some different stages of being put together into functional myostatin, and SRK-015 goes after kind of the early phase, like upstream in the factory production line. And the thought there is if it gets it earlier, that it could be more effective in knocking it down. So so that's been shown in mouse models to work, and so we'll see what happens if they take it into the clinic over the next few years. Then there's a lot of excitement around MRI as a tool for predicting, visualizing how FSHD is affecting the muscles, and then whether we can use that information to predict how somebody might progress. And that's really important, obviously, for individuals who might want to know, what's my prognosis, but also super important for clinical trials where, you have basically a year to show that a drug is going to change the trajectory for somebody with FSHD. So if you have a group that you can tell they're on the path to develop more weakness over the coming year, then you have a better chance of being able to show that your treatment can really change that trajectory. As you all know, like, you can go through periods where you're just kind of stable or not changing a lot. And if you had a group of people like that in a clinical trial, you might try treatment that would actually work, except you'd never show it in that particular population because they're just at this kind of stable stage. So you want to find people who are on the path to getting worse over the next year, and then saying, can we give them a drug and stop the progression? So MRI is, magnetic resonance imaging. You can take pictures and do 3D reconstructions of the muscles throughout your body, throughout whichever area the MRI is scanning. And it's pretty cool because we know that in FSHD, the muscles start to fill up with kind of scar tissue and fatty infiltration. And that shows up like as a cloudy, increasingly white signal on the MRI. And you can, if you scan like an entire leg, you'll see like spots of muscles that are affected here and there. And if you think about like most tests, like, let's see how it changes your walking speed. Well, your leg uses all of these, dozens and dozens of muscles to walk. And if 3 of them are affected by FSHD, you might be able to compensate. Your muscles are going to like compensate, and it'd be, it might not change your walking speed significantly. But on an MRI, you could see like, oh, these muscles have continued to get more fatty infiltrated, and then there's another one that goes. So having that kind of objective ability to look at muscle over the course of a clinical trial is super, super important. And what Sylvia Blemker has done with Springbok is they used AI because there's a huge amount of data. You're taking tons, little thin slice layers of MRI from people. There's just figuring out which muscle is which is a huge task. And then you're looking at it over, month over month over month in different patients, trying to find a change and trying to find patterns. And so they have shown that they can successfully use AI to analyze this MRI data and predict which patients are going to be developing muscle weakness more rapidly. So that's super important. Next here, we're looking at something called gait-based biomarkers. So I think, again, relating to your own personal experience, if you have something like 93% of people with FSHD have some it affects their walking in some way. It might be very slight, or it might be very dramatic to where, severe, where you can't really stand well or walk independently. So, but it's very widespread. And I'm sure you also know that as you develop weakness, that the way you walk changes, right? And you're compensating there. You're like, with your balance, and your whole body is actually compensating as you walk. So if you had a test, like how long does it take for you to walk 10 meters? Like, you might, over a period of years, be able to cover that distance in the same amount of time. I don't know, 6 seconds or 7 seconds or whatever. But the way you do it will change. You might need to move your body more in certain ways to compensate for the weakness that's developing in your hamstring or in your tibialis or something like that. So just looking at the time is a very crude measure. If you can look at how your gait, the way you're walking changes, that might be more sensitive. And so this group in England used sensors that are attached to your body. And I don't know if it's just the legs, or because they're also saying like your torso and your upper body also change, and there's all sorts of compensations that go on. And if you can have sensors that detect those changes and analyze that, again, probably with AI, that we should be able to have a much more sensitive way of measuring functional change in people. So I know I'm taking way too long here. Okay, I'm going to skip because we're going to get back to this biomarker thing. There was a really interesting presentation by Emma Weatherly from FSHD Global Research Foundation, based on experience of Bill Moss, who's the founder of FSHD Global Research. They're in Australia, and Bill has FSHD. And over the last few years, he experienced a major, like a medical crisis, maybe a breathing related or whatever. He ended up in the emergency room and suddenly just had a first-hand experience of not having a palliative care plan in place. So palliative care, people often associate it with end of life, and it's certainly relevant at the end of life, but it's also at any stage along the way where there's not a treatment or intervention that will change things, but what you really want is just to be comfortable, just to have your pain addressed, just to have your daily life, quality of life things addressed and made easier for you. All of those things fall under palliative care. And right now we don't have a medical guideline on palliative care for people with FSHD. So Emma is going to be leading an effort to develop such a guideline. And now they also presented, Dr. Statlin presented data from the Avidity clinical trial, which just pretty much concluded its phase 2 and is about to launch a phase 3. And just as a quick primer, oh, are we out of time? We're almost out of time. Sorry. This is a schematic of a cell, and we have in here DNA, which reads out a strand of messenger RNA, which leaves the nucleus, and it gets into some machinery that takes that genetic code and turns it into a protein. So what Avidity does is it kind of blocks this mRNA. It has a molecule that recognizes it, gloms onto it, and destroys it. Or other things come and destroy it once it gloms on. So, and that's all I'll mention at this point, so just as a reminder. And their drug is attached to something called a transferrin receptor, which is found, the transferrin receptor is found on the surface of muscle cells. And so an antibody to it you inject it, it goes and it recognizes, it binds to this receptor on muscle cells, and it gets inside. And when it does that, if it's attached to this anti-DUX4, let's say this oligonucleotide it's called. But anyway, it's this thing that is going to find the DUX4 messenger RNA, bind to it, and help neutralize it. So that is the basic design of this drug. So it's a fairly large molecule, so you can't take it as a pill. It's given through an infusion, and it has been in this phase 2 trial, which involved 5 infusions. I won't go into the details of who was what the inclusion criteria were, but people were administered this drug over approximately a 12-month period, and the data have been completed. And we just had a webinar yesterday presenting the data. I'll just quickly speed you through. The main point here is orange is the group taking the drug, which is called Delbrax, and gray here is the placebo group. And everywhere you see, like here in the reachable workspace, you see the group on the drug is improving. This is where nothing changes. So they're improving, and people on placebo are slightly declining. With quantitative muscle testing, again, the group on Delbrax is either stable or not doing so bad, doing better than the placebo group. And the 10-meter walk test, this way towards 0 is better. It means you're the faster, or is it change? I think it's the change over the period of time. So this group has not changed much, whereas the placebo group has increased in their amount of time it takes them to do the 10-meter walk test. Again, here you can see the oranges are in the improvement side across all of these measures: pain, overall health. These are questionnaires, global impression, and they're saying, well, what is a change of 2 to 3 seconds, which is what we're seeing in the placebo group? Their walk slowed down. What does that mean in real life? And it can mean anything from someone being able to walk independently to needing an AFO, or if someone's already using an AFO, it might mean they need more support, may need a cane or a walker, and so on. So these are significant for the patient. And the other exciting news from Avidity is that they looked at proteins, molecules in the blood of the people participating in the clinical trial. And what they're looking for is what's called a biomarker, which is a biological marker that represents the disease process. And for FSHD, one type of biomarker you might want is one that tells you how active the DUX4 is in a person's body. And that biomarker could come from a biopsy. And some of you have had that where you just actually take a little piece of your muscle and analyze the DNA and other proteins in there. Or much better be if it came from the blood, and you don't have to give up any muscle, you just draw some blood. And also it's more ideal, because in a muscle, it's hit or miss. Maybe you'll get muscle from an affected muscle, but you might get it from a healthy piece of muscle, and that wouldn't tell you too much. Whereas in the blood, if there's a muscle that is being affected by FSHD at a given time, and it's shedding something that gets into the bloodstream, then you can tell somewhere in the body of that person there's a muscle that's affected by FSHD. And so that is something that many people have been looking for, and it's really exciting that Avidity thinks they have found one. So I just skipped through all of this. I think you don't need any convincing that having a blood test would be better than having a muscle biopsy. And what I show here are 2 graphs, one that's showing one of these biomarkers, which is specific for DUX4. It's called cDUX4. And that biomarker, okay, what we have here is in people on Delbrax, the level of that biomarker went down, whereas in the control group, it stayed steady or went slightly up. So that's a nice indication that this biomarker really shows you whether the drug is having the intended effect or not. There's a second biomarker called creatine kinase, which some of you may have heard about. Many of you, when you're diagnosed, might have had a blood test to look at creatine kinase. So this is a protein that is produced when muscles are damaged. You will see it in bodybuilders after a heavy workout and stuff, where the muscles breaking out, you'll see elevated creatine kinase. And you see it in people with muscular dystrophy. And so here too, so that this is not a DUX4 specific biomarker, but it is a non-specific biomarker of muscle damage. And what you see again is in placebo, the levels of creatine kinase are kind of up at a steady level. And in people on Delbrax, it falls. So those are two, that's what you want to see in a drug that is actually working. So these are good indicators. And now Avidity is planning a phase 3 trial. I can't tell you too much about it. I know that many sites are preparing for it. It's probably going to be a few months before they are all ramped up and ready to start running the trial. But I think in the meantime, we are encouraging people to get on the list at sites that are having them, because they're going to start going through those lists and screening individuals to see if you are eligible for this trial. So I'll just, and I can just end here because I'm way past my time. Thank you.